Thanks, Ralph, for the invitation to give you guys a bit of an overview of what it is we're up to and how we use Compound Discoverer. So I'll just start off by giving you all a bit of a background on our project, going over how we set up our experiments and then how we use Compound Discoverer in our work. And then I'm going to show you a few preliminary results. They are hot off the press, so please bear with me, and how the utility of various tools in CD has helped us in our untargeted analysis, and especially the fact that we're being already able to see interesting differences in the uptake and excretion of compounds by different cell lines. So the aim of our project is to try to infer transporter substrates from metabolome data and use that together with transcriptome and proteome data. So the way we're going to do this, or we are doing it, is by using metabolomics, untargeted to begin with, measuring the compounds that are present in serum that are taken up by cell lines over time. And then eventually we'll compare these, or correlate these to the expression levels of the various transporters using our mRNA sequencing data or protein expression data. And the idea is that that will allow us to infer some of the substrates, at least for some of these transporters, not only natural substrates but also xenobiotics, so drugs and other synthetic compounds. So just to start off with, how do metabolites and drugs get into cells? So it's wrongly thought that they just pass through or they just diffuse through the phospholipid bilayer of cells. But that, of course, is wrong, because otherwise your cells would be full of all sorts that's around in the atmosphere or anything nasty that you eat. To a certain extent, yeah, you are going to get nasties into your cells. But the way they actually get into your cells is through transporters. And drugs also use transporters to get into the cells. So it's no surprise then to find that the majority of pharmaceutical drugs, marketed drugs, are very similar in their structures to a lot of the natural compounds that are out there, or a lot of the natural metabolites that you or I have in our bodies. And just to confirm this, we have done, or my colleagues have done some previous work before, looking at the structural similarities of the recon-2 metabolites with the marketed drugs using various encoding algorithms. In this case, it was the MAX encoding algorithm. And I find that indeed there's some sterols that are very similar to various steroids and various CoA derivatives here at the bottom. But even more so when they looked at the Tanimoto similarity between drugs and the recon-2 metabolites, they found that 90% of these drugs had a similarity of 0.5, a Tanimoto similarity of 0.5 or greater to the nearest metabolite. So at least we're not going completely haywire in what we're doing. So what are the proteins that transport metabolites into and out of cells? So there's two main ones. We've got the SLC series, which generally take metabolites into the cells. And we've got the ABC transporters, which generally export things out of the cells. And of course, then we've got various pumps, ion channels, and water channels. And there's around 400 SLC transporters, so the ones that take stuff in, and around 50 ABC transporters, which are very important in cancer drug discovery. And further work we've done looking at the expression of SLC transporters in human tissues has shown that transporters are very widely expressed in the human tissues. So we used here the Gini coefficient, which is normally used in economics to look at the distribution of wealth in countries. So a Gini coefficient of 1 means that everything is very unequally distributed, as is in most places. And a low Gini coefficient is more like a communist country. So in our case, we found with the SLC transporters that there was a number of SLCs that had a high Gini coefficient. Is this working? I don't know if it's working. Anyway, you can see it on the top. There's SLCs with a high Gini coefficient, so that means that they are pretty much expressed on 1 or 2 or 3 tissues. And at the bottom, we've got the ones that are expressed pretty much everywhere. And lo and behold, we find, if we look at the right-hand side here, SLC01B1 is a transporter that's mainly expressed in the liver, and it's responsible for statin uptake. And at the bottom, we see various SLC25s, which are mitochondrial transporters. And of course, all your cells need mitochondria to produce energy and ATP. So this is all making sense. So then it comes to the work that I have to do, which is measure the uptake and excretion of serum metabolites in human cell lines from different tissues. What I'm showing you today is just for 4 cell lines. They are all from different tissues, but as we go along, we're increasing the number of cell lines that we have to cover as many tissues as possible, and therefore be able to try and figure out which transporter transports which metabolite and therefore which drug. So how do we do this? We grow the cells, we pellet the cells, we resuspend them in serum. So this is human serum, it's pooled serum. We've bought, I think it was about 2 litres of it and aliquoted it in lots of little aliquots. So every time we add the serum in here, leave the cells for a certain amount of time. In our case, it's up to 1 hour. We centrifuge them, take the supernatant, extract the extracellular metabolites, and then go on to the intracellular metabolites and RNA. So most of the stuff we're doing is with the extracellular metabolites, and it's what I'll be showing you today. So how do we acquire the data? So it's mainly based on a Nature Protocols paper by Rick Dunn and colleagues. So we're using a QE+. It's not our QE+, it's some colleagues that have very kindly let us use it with an LC. We're using a 15-minute chromatography separation, reverse phase, C18 column. We're doing MS1 and MS2 data acquisition with data-dependent manner using the top 5 and various master charge windows in there. We're doing all the pre-processing in Compound Discoverer, and then the data analysis and visualization. We're using 9, and we're using various R scripts and Python within that. And the workflow that we're using is pretty much identical to one of the templates that's, if not completely identical actually, I can't remember now, I've changed so many things as I've gone along, but it's based on one of the templates. I've changed various parameters in there. Yeah, I don't think there's much else to say on that. So for us, we're doing a lot of batches with a lot of samples over time. So it's very important. The nodes that do the retention time correction and QC-based area correction are extremely important to us. So here you can see it is doing a pretty good job of the retention time corrections here. And again, for the QC-based area correction, so you can see, well, you can't really see, but various runs there between September and November. So there are going to be variations, but there's not much we can do at the moment because we don't have our own instrument. But it is doing a really good job of this. One minor niggle I'm finding is that the plots, you can't really see them very well. It's a minor niggle, so hopefully the software guys can hopefully do something so we can change the font sizes, etc. on these kind of plots. I know you can do it with one of the other plotting functions in Compound Discoverer, but with these ones, I've just had to use PowerPoint. So it'll be really useful to have that. And the MZ Cloud, MZ Vault, and fish scoring has been extremely useful for us for the improved identification and confidence in the identification. Again, we could do with being able to change the font sizes, etc. But for us, it's worked really, really well. It would be quite useful in the results table if we could have INCI or SMILES identifiers so that we don't just get the names. Various compounds have various different bizarre names, which makes things quite challenging. So I hope the software guys might be able to integrate that in there. Please. It seems to have decided to stop working. Okay, I'll just do it on the computer. And yeah, as Ralph was saying, our sample sizes are quite big. And it is becoming a bit limiting. So I know there was a bit of a discussion earlier, what can we do to make it faster? Yeah, any help would be most welcome. I mean, I don't think it's our computer, it's a pretty meaty piece of kit, and it's not the internet connection. I don't think you can get much higher than 1 gig per second. So any ideas or help would be much appreciated on that. Because as you can see, when we've got 8 cell lines, it's nearly 3 days. Eventually we're going to have up to 50 cell lines. The time per sample? Is it going down? Well, hopefully. Yeah. So I should do 50 runs. It'll only take 2 hours then. Oh, that would be nice. And then, yeah, just some results with the number of features and compounds. We're only running in ESI+. Again, it's due to the fact that we don't have our own system. So we get most of the metabolites that we're looking for in ESI+. So at the moment, this is good enough. It is a bit, well, I don't want to say disappointing, but we only get sort of 400 odd results in MZ Cloud, but these are good results. And of course, the database is growing day by day. So it's not really a criticism, but if one was here. And then, so some of our PCA plots here. So you can see over time, we've got, if you look at the purple squares, those are our QCs here. And we can see differences already in PC1 for cell lines and also over time. So over here we've got, I think it's the PANC1 cell lines, 60 minutes versus 0, which is around here. So things seem to be working. And on the right hand side we've just got time 0 versus time 60. And we can see very good differences between the cell lines and also over time. On the univariate stat side of things, again we can see differences in the number of compounds that are consumed, the number of compounds that are secreted. They're different between the cell lines, and also the magnitude of these are different between the cell lines, and it's also increasing over time. But what we're to try and narrow down what it is that's changing over time in the cell lines and what's unique and different between the cell lines. For now, we're just looking at 60 versus 0. So we did these other time points just to see, do we already see something at 15 minutes? Do we see something at 30? Is it even bigger at 60? And again, lots of numbers, but you can see there's quite a lot of compounds that are consumed, quite a lot that are secreted, a lot that are unique to each cell line, and quite a few that are common across all the cell lines and between the various different cell lines. So to start off with, I've just started to look at what's unique in each cell line, and then also what's common across all the cell lines to try and figure out, okay, so there's a particular metabolite that's consumed just by, or much more by one cell line compared to the rest. And then eventually we're going to look at the RNA sequencing data or the protein expression data, and hopefully that will show us, okay, well, this transporter is much more highly expressed in this cell line than the other, which explains why, so that's probably a substrate for this transporter. Well, that's the ideal vision, eventually. So, thankfully, we see glutamine is consumed by all the cell lines. So at least they're not dead, they're actually alive, they're doing what they're meant to do. And it's a pretty good match on MZ Cloud and also with the fish scoring. Sorry, I've got a dry throat. And we also see large differences in the amount of glutamine consumed by the different cell lines. Particularly, we see the PANC1 cell line consuming a lot more glutamine than the other cell lines. So again, hopefully, when we look at the sequencing data or protein expression data, we'll see large differences in the expression of various glutamine transporters here. And lactate, again, that gives us some confidence that the cells are indeed alive. Cancer cell lines tend to secrete a lot of lactate. And again, we see that there are differences between the cell lines, and again, the PANC1 cell lines seem to secrete a lot more lactate. This might be all the way down to a different metabolic network in there, leading to a higher consumption possibly of glucose and secretion of lactate. But we're not interested so much on the excretion, we're more interested in the uptake. Interestingly, we found a number of nucleosides, in this case it was cytidine, that are excreted by the cell lines. Not really sure why they would do this, but I imagine it's maybe down to the fact that they're in exponential growth phase. They might be overproducing nucleosides, and rather than letting them become toxic, maybe they're secreting these to prevent any toxicity here. Compound discoverer did say that this was cytarabine, which is a chemotherapeutic drug, which, as you can see, is very similar to cytidine, same mass to charge ratio, etc. So you have to use your brain a little bit, and it's unlikely that someone taking a chemotherapeutic drug is going to be donating blood for these purposes. So it's most likely to be cytidine that the cells are producing here. And then we see some, of course this is from humans, the serum. So we do see a number of drugs, legal and illegal, showing the legal ones for now. But for example, Gabapentin, we saw this is a drug for the treatment of epilepsy, but also for the treatment of various pain conditions. And again, we see different uptake of gabapentin by different cell lines. So I'm guessing the PANC ones must be in a little bit of pain because they consumed a lot more compared to the other cell lines. And again, when comparing the MSMS spectrum to MZ cloud, we had a really good similarity match there. So we're nicely confident that this is what it is, what it's telling us it is. And then another drug, I think this is the most exciting bit, finding what various drugs in the serum that the cells are taking up or not. Trazodone, an antidepressant drug. Again, 2 cell lines consumed it, 2 cell lines didn't. Good match on the MSMS spectrum. We don't know what the transporter for this is. It's probably the serotonin transporter. Again, if we look at the expression data, we'll hopefully see differences in the expression of these. And then, yeah, just citrulline. Again, 2 cell lines consumed this, 2 didn't. And sphingosine. So I hope I've convinced you that we've got a nice protocol here developed to measure the uptake and excretion of serum metabolites by LCMS. We're identifying metabolites both with biological significance and also pharmaceutical compounds that are present in the serum, and we're also seeing differential consumption and excretion by the cell lines. So our next steps are to increase the number of cell lines. Hopefully Compound Discoverer will be faster, like we said. We're hoping to increase the breadth of the detected metabolites, but one step at a time. We'll go on to a negative mode, as well as hillic chromatography, and compare these results to the transporter expression, like I said before, to infer the various substrates for the various transporters. And I find that Compound Discoverer is a great platform for the data pre-processing side of things. Like I said, could do with it being a bit faster, but there are limitations to this, as we discussed this morning. Really would like annotation with unique identifiers and some improvement on the plotting capabilities. And lastly, I'd just like to thank everyone in my group, especially Farah Mughal, who's been doing all the cell culture side of things, Professor Kell and Dr. Philip Day, who are my advisors, and Steve O'Hagan, who's helped a lot with the 9 workflow. Thank you very much.